Okay, so uh, hello everyone. Uh, we're going to start our first unit today called Peptides and Proteins. And this involves 4 units essentially within this part. And we're going to be looking at the properties of amino acids. We're going to look at the synthesis of how these peptides and proteins were maybe made prebiotically, and also talk about enantioselectivity. So the first thing that we'll be talking about is natural amino acids and properties. So as you can see, the outline of this presentation is divided into 4 parts. So this first part will talk about natural amino acids and properties, and then these other sections, like how are amino acids formed, chirality in amino acids, and chiral amino acids as a catalyst for desugars, we will do those afterwards. Okay. So first of all, why do we care about proteins? Now, if this were, a face-to-face classroom, I'd be looking for hands up. But because this is a short recorded lecture, I will just tell you some very fascinating things about proteins. So proteins, catalyze the biochemical reactions in our body. Okay, so they're responsible for all these metabolic pathways and all the different functions that our cell and body uses ultimately. So proteins, most biochemical reactions. And back around 20 years ago, at the turn of the century, scientists discovered that we had roughly 30,000 genes. Okay, so scientists did a large amount of sequencing, and it was actually surprising that we discovered 30,000 genes. A lot of people estimated that we would have hundreds of thousands or even a million different genes, because I guess researchers thought that we are just such complex creatures. However, we have 30,000 genes, which is the same number of genes roughly that monkeys and chimpanzees have. But nevertheless, what this means is that there are roughly 30,000 different proteins. And while we do know and understand the function of a lot of these different proteins, the vast majority of proteins, we still don't fully understand their function. So therefore, that's why we care about proteins. Some other good reasons why proteins are important is that proteins are excellent drug targets. So as I talked about in sort of that introductory lecture, most pharmaceuticals target enzyme active sites and inhibit the function of a protein. So by understanding the function of the protein, you can actually design very good drug targets. As we will learn about later on in the course, proteins are not the only drug targets out there. Other biomacromolecules can also be very useful drug targets in pharmaceutical drug discovery. So most pharmaceuticals target enzyme active sites. Furthermore, proteins can also be used as therapeutics. Okay, so we can not only use them as drug targets, we can actually engineer them, we can actually make them, and they can actually be big pharmaceutics, big drugs. Okay, so I'll just highlight sort of one very, very brief example. Here would be your cell membrane, and on the outer surface, this is inside of the cell, this is outside of the cell. Some cancer cells exhibit or express various proteins on the outer surface. So this is a specific protein. And what antibodies do, antibodies have very specific and high affinity for proteins. And therefore, antibodies can be used to target specific proteins that might be involved in cancer, for example. Okay, we will touch upon that later on in this unit. Okay, so there's a little bit of a reason why we're going to be studying proteins. Okay, so a little bit of background on proteins. So proteins are perhaps the most versatile organic molecules in nature. And I like to think of proteins as organic molecules. You'll see in a minute. But as you've learned in biochemistry and other courses, these proteins have various side chains that really characterize how they function and what their properties are. So a lot of this slide here contains review from biology and second year biochemistry, or maybe third year biochemistry, but it's always good to review it. So proteins are made of a combination of building blocks to create unique macromolecular primary sequences. So these are called amino acids. Amino acids contain various functional group side chains. Proteins are relatively rigid molecules, which are ideal for creating binding or enzyme active sites. Furthermore, proteins have the ability to fold into very complex three-dimensional structures that govern its biological activity, and they can also interact with other large proteins to form macromolecular complexes. So, an example of a good macromolecular complex would be something like the ribosome, which has lots and lots of different proteins together, in addition to RNA to catalyze protein synthesis. Let's look a little more closely at the structure of amino acids. So at the central carbon, which I'm highlighting here, we have an alpha carbon, and attached to it we have a carboxylic acid or carboxylate group and an amine group. Okay, what I'm showing you here is the Zwitter ionic form, which is present at neutral pH. I'll talk more about that in a little bit. But they're called amino acids because this is an amine group and this is an acid group. It's an asymmetric carbon. So as you can see, this carbon has 4 different substituents, and the ionic properties of the side chain, which is the R group, influence the physical and chemical properties of the amino acids and therefore the protein once it's embedded within it. Here on the right, I'm just showing you that because these are chiral molecules, they can exist as 2 different stereoisomers. So let's take a closer look at that. Okay, so some of the properties of amino acids. So before we talk about the stereochemistry of amino acids, I want to take a look at a well-known metabolic molecule. It's a very simple 3-carbon molecule that you've seen before in biochemistry, glyceraldehyde. Okay, so this is involved in glycolysis. Now, so researchers, what they did a long time ago is that they isolated the 2 different stereoisomers of glyceraldehyde. So you can see here there's a central carbon and there are 4 different things attached to it. Okay, and at the bottom here we have another glyceraldehyde molecule, 4 different substituents, but you can see here that it is the mirror image. Okay, so back a long time ago, the way they characterized this was that they took circularly polarized light and they asked the question, how does this molecule deflect, or how is it rotated after it interacts with this chiral compound? So when circularly polarized light interacted with glyceraldehyde here that I'm circling, they noted that this rotates the light to the left. And this is often known as levrotatory. Okay, so the L comes from levrotatory, and that's why L-glyceraldehyde is called L-glyceraldehyde. Conversely, the other stereoisomer takes circularly polarized light rotates to the right, okay? And this is known as dextrotatory, okay? Dextrotatory. Okay, so that has the prefix D. That's why this is called D-glyceraldehyde. So these are some sort of historical nomenclatures that researchers would use. And if you take a look at glyceraldehyde, you can see that it's kind of similar to amino acids because you have a central carbon, you have 4 different things attached to it. So if you take something like alanine, which is an amino acid where one of the R groups has a methyl group here, this actually will rotate light to the left, and this is what is known as L-alanine. Now, this is where there's a bit of confusion with how these amino acids are actually, or how they're actually depicted in terms of their nomenclature. So later on, organic chemists actually designated absolute configuration, okay? And you guys learned this in 2nd year. This is where you take the central carbon here, and what they've done is they've oriented the hydrogen to the back, and then they use the pre-Ingold rules to define stereochemistry. So just to recall, lowest priority hydrogen goes to the back. The highest priority in this case is a heavier atom that the carbon's attached to. So this would be the oxygen. The second priority would be either this carbon here or this carbon. Now, this carbon is bonded to 1 oxygen and 2 hydrogens, whereas this carbon here is bonded to 2 oxygens because if there's a double bond, it's an aldehyde, and 1 hydrogen. So therefore, this gets higher priority, that's number 2, and this gets priority number 3. So if you actually go from 1st, 2nd, and 3rd, you'll notice that this goes counterclockwise. And you've learned that if it goes counterclockwise, this is known as S configuration. So L-glyceraldehyde is actually also known as S-glyceraldehyde. Conversely, D-glyceraldehyde, you can see that if you add your priority groups, 1, 2, 3, you will now go in the clockwise motion. All right, now this is where it gets just a little bit confusing. L-alanine, okay, does equal to S-alanine, all right? So, you know, we can easily put 4 there as our priority, and we can add 1 for the amino group, 2 for the carboxylic acid group, 3 here. So you can see here that this is S, that's S-alanine. Now, all natural amino acids, okay, we live in an L amino acid world, or racemic world. All natural amino acids are S except for 1 amino acid. Except for this one here. So here I'm just going to draw what that amino acid is. If you don't know it, it's fine. We haven't talked about it yet. But it's one of the sulfur-containing amino acids. Okay, so again, hydrogen gets 4, NH2 gets 1, but this carbon is now bonded to a sulfur, and sulfur is now heavier than 2 oxygens, so it actually trumps the 2 oxygens. So now this gets a higher priority, and the carboxylic acid group is now priority number 3. So this actually rotates in the R configuration. So while L-alanine is equal to S-alanine, and all the other L amino acids will equal to S, whatever that amino acid is, the exception is that L-cysteine actually equals to R-cysteine. Okay, so all natural amino acids are S except for cysteine. Okay, and there's another exception, and glycine, because glycine has R equal to H. Okay, so as soon as you have R equal to H, that would mean that you lose that stereo center. All right, so those are just some of the properties of amino acids. What I have here is a picture of just all the different amino acid side chains. So these are the 20 very common side chains that characterize amino acids. I don't expect you to know or I don't expect you to memorize all these amino acids, but you should be familiar with them and try to understand them from a fundamental organic chemical point of view. So they're characterized into different blocks. You can see you have acidic, you have basic, you have polar hydrogen bonding and sulfur containing. Okay, so acidic ones you can see here have these carboxylic acid groups. So the pK of those are roughly around, between 3 and 5. Here you have a tyrosine group. This hydroxyl group is also acidic. You have basic amino acids here. Okay, so these basic amino acids have the ability to pick up a proton. All right, there actually should be 2 here from the textbook. And they're also characterized whether they are aromatic or not. Okay, so these are, these ones here are obviously aromatic amino acids or aliphatic. So aliphatic are hydrophobic, that are carbon-containing amino acids. We also have polar hydrogen bonding ones. So these are amides here. So these can hydrogen bond with a variety of different hydrogen bond acceptors. Okay, so that's asparagine, glutamine. This is serine, that's hydroxyl group here, can hydrogen bond. This is threonine that also has a hydroxyl group. Here you have methionine, which is sulfur-containing amino acid, and cysteine. Cysteine is actually kind of special. We just learned about its absolute configuration. It's actually R, but cysteine also can participate in polar hydrogen bonding, but as we'll learn too, the cysteine is actually a very good nucleophile and can actually form disulfide bridges, among other things. And then we have what is characterized as flexible and inflexible linkages. So glycine, as you know, has 2 hydrogens here. And because those are so small, glycines can actually fit in places where the peptide chain might need to really fold or kink excessively to form a complex structure. In contrast, amino acids like proline have a 5-membered conformationally constrained ring embedded within it, and this prevents the peptide from actually kinking and adopts a more sterically constrained structure. So anyways, that's just a little bit of a highlight of some of these amino acids. We'll definitely talk about them more in the coming weeks. I've also characterized the amino acids here. Again, you can think of amino acids as being neutral, okay? Or you can think of them as being acidic or basic, okay? And this, of course, depends on their pH. All right, so what I want to talk to you about is that here is an amino acid and a physiological pH, which is pH around 7, okay? So that's within our bodies. Because the pKa of this carboxylic acid is around 2 to 4, what this means is that this amino acid is actually going to be deprotonated. This carboxylic acid is going to be deprotonated at physiological pH to give us our carboxylate. So this is now negatively charged. In contrast, this amine group will actually pick up a proton because the pKa of an amine that is protonated is around 9 to 10. So this is now above our environmental pH. So this will be protonated. So this is known as a Zwitter ion. Okay, so it's negative and positively charged. And depending on the environment, okay, you can have little local microenvironments. If there's a base around, well, that base would be around and that would remove that proton from the amine, giving you now a negatively charged structure. In contrast, if you have an acidic environment, this carboxylic acid is going to remain protonated, and you will be now left with something like a protonated amine. So this will be positively charged species. So depending on the types of amino acids present within the protein can dictate what is known as its isoelectric point, the pH at which the protein is neutral, and it can also tell you a little bit about some of these properties and charges at various locations throughout the protein. Okay, so yeah, so the last thing I want to do is before we start talking about more chemistry and to finalize this unit, I want to just go over the review and go over the mechanism for drawing arrows. Okay, so what I'm going to do is I'm going to use predominantly the paper for these lectures, but once in a while I do have a whiteboard behind me, and it is sometimes nice to see a face, right? So I'm going to go through this mechanism and just sort of highlight what's important. Bear with me while I turn my camera. All right. So, all right. So this is the last question on that. Okay. So, hi everyone. Hopefully this is clear. So draw a plausible arrow pushing mechanism for the following tautomerization reaction. All right. So let's do that. So let's draw the structure. So take this amide with catalytic acid, HA, and it will tautomerize to that species there. So the question is, how does that happen? How do you show that mechanistically? Okay, so the important thing to know is that we are under acidic conditions. How do we know that? Well, we have an acid present right here, HA, that is our generic acid. Okay, so we don't have any base present. So what we're going to do is we're going to first activate this carbonyl group. So the lone pairs will react and pick up a proton, releasing the conjugate base. So what this will look like is a structure that's now positively charged. And then what you will have left is the conjugate base, okay? So remember, this is your acid, this is your conjugate base, all right? Now, in order for us to go from here to here, a couple things have to happen. We need to form a double bond. We need to lose this double bond here, and we need to lose a proton, okay? So the way that I'm going to do this is I'm going to say, well, this positive charge oxygen is, we're going to break this bond, but that's going to be assisted by the nitrogen coming down and the electrons, the pi electrons moving up to the oxygen. Okay, so now I have this structure here. Nitrogen now has 4 bonds. The nitrogen is positively charged. And now our conjugate base can come and pick up a proton from the nitrogen to give us our final product. So the acid is regenerated. And again, this is in catalytic amounts, so that can be used to recruit another starting molecule. I just want to emphasize and highlight things not to do. So recall and remember that electrons always, or arrows denote the flow of electrons. Okay, so here I have a lone pair, okay, and that's going to go to an atom or pick up a proton. Okay, so what you cannot do, and I see this every year, is if you have some kind of proton here, you cannot, for example, have the proton attacking the hydrogen to give you this desired structure. Okay, this doesn't make sense fundamentally. First of all, the proton doesn't have any electrons, so it can't move electrons to here. And furthermore, this oxygen already has a full octet, so it can't accept any more electrons. So make sure that you draw the arrows in the right orientation. Okay, so don't do that. All right, so that concludes this first portion of the lecture.